We would like to start off by thanking you for the opportunity to present our innovative new use of triplex technology. Our mission here at GeneHuman is the alleviation of genetically rooted disorders through triplex DNA technology, and male pattern baldness is the first step. Male pattern baldness affects up to 80% of men by age 80. Today we will explore the androgen receptor gene's role in male pattern baldness and our strategy to minimize its impact. With the help of your funding, we plan to use triplex technology to specifically target the functional variant of the androgen receptor gene through site-directed deletion and oligomer pairing. Androgenic alopecia, more commonly known as male pattern baldness, is the most common form of hair loss worldwide, and it currently has no cure. The emotional stress of hair loss can have adverse psychological effects, prompting a 2006 study on the issue. The Erasmus University study noted that average reactions to diagnosis of androgenic alopecia range from mild to severe annoyance, and an Old Dominion University study cited the effects as serious as body image issues and struggles coping. Our company understands the enormous potential of genetic engineering and triplex DNA technology, and feels that no one needs to further endure the emotional stresses of male pattern baldness. Currently, male pattern baldness is most commonly treated with finasteride, a medication that blocks the enzyme 5-alpha reductase. However, this medication has numerous side effects on male primary and secondary characteristics that are linked to low levels of dihydroxytestosterone. Fortunately, male pattern baldness and many other maladies to which our research and technology could be transferred, such as prostate cancer, have been linked to the androgen receptor gene malfunction.
The gene we are looking to target to take control of the tragedy of male pattern baldness is the androgen receptor gene. A University of Melbourne study compared the genetic makeup of young bald men and a group of older balding men versus a control group of men with no apparent baldness. The study first displayed that the short allelic repeat of the GGC sequence within the androgen exon 1 site of a functional variant was important in the receptor's hyperactivity. Also, the androgen receptor gene STUI restriction site on the X chromosome was detected in 98% of the young men and 92.3% of the older men plagued with baldness in the study. The STUI marker was found in other non-bald men, but it was not in linkage disequilibrium with the GGC genetic mutation. The presence of the GGC sequence leads to a disruption in the regulatory region, causing an overexpression of the androgen receptor gene. Since the gene produces a steroid receptor, overproduction will increase the sensitivity to the androgens in affected cells. This will lead to an increase in the transcription and translation of androgen-responsive genes that contribute to male pattern baldness. Mutation in the androgen receptor gene is a key factor contributing to the polygenetic expression of male pattern baldness. Mutations in the androgen receptor gene are the only known factor of male pattern baldness discovered thus far and account for 40% of heritability. If a male is tested positive for the GGC and STUI restriction polymorphism, and they also have a family history of baldness, then there's an 80% chance that they will also develop the trait, representing the significance of the gene.
The genetic pathway we are targeting is a pathway that contains the androgen receptor produced from the androgen receptor gene. The androgen receptor is part of the pathway in which cholesterol is transformed into androgens, illustrated in this diagram. In this pathway, the androgen receptor then binds to the androgens testosterone and dihydroxytestosterone, creating an androgen receptor complex. This complex then attaches to DNA and regulates the expression of androgen-related genes. Finally, we will explore our plan to use triplex DNA technology to diminish the frequency of male pattern baldness. Triple helix forming molecules recognize oligopyrimidine and oligopurine sequences through hydrogen bonding interactions. Regulatory control of the androgen receptor gene may be accomplished by triple helix construction using a third-strand oligomer or a triplex oligomer pair. The third-strand oligomer technique uses complementary oligomers to hybridize to a segment of a double-stranded nucleic acid and form a triple helix structure. We plan to use an oligomer compound to bind with the GGC repeat sequences found in the overexpressed androgen receptor genes. We will then modify this triplex-forming oligomer to be covalently bound to a topoisomerase I inhibitor that can facilitate DNA cleavage to the specific GGC sequence. This is crucial since androgenic alopecia is a result of a disequilibrium between the GGC sequences and the STUI restriction site. Excision of these sites will help to regulate the overexpression of the androgen receptor. So now we ask you to look into the depths of your heart and pockets to fund this sensational research so that we may eliminate the shameful prevalence of male pattern baldness.